right, so let me just go through this. This is an idea that's a little simple. It's simple. I haven't worked it all the way through yet, so there might have some errors in it. Um, um, but the details are complicated. I don't want to go through all the details, so I'll do my best. So it's sort of something I stumbled along. I realized that this could be an issue with the scale problem. And the scale problem is we're observing an object that's at a different scale than when we learned it. And we seem to be able to do that very rapidly, quickly. It doesn't take any time. It just happens immediately. So it seems like certainly not effortful. And this is a potential solution to that problem. So it has to do with grid cells and how they work and how cells work in the brain. So it's a very neuroscience-y solution. Before I go through the neuroscience, maybe I'll just describe the basic idea. What we're trying to do, our basic models are we have some location in a reference frame, and we're pairing it with some observed feature, which could be a curvature or something else. And when we're correctly on an object, as we update our locations, it points to the correct new feature, and a feature then can narrow down our locations. The idea here is that as you're moving your sensor or your body or whatever, the thing that's moving, and you're updating your location, it actually sweeps through a series of locations over and over and over again in a linear line. So if I'm moving in some direction or my finger's moving in some direction, it'll sweep through a series of locations along that line of movement about 10 times a second. And you would be able to match the observed feature early or late, and it would know that. It would say, yeah, I recognize it was in the wrong location. It's a little too further away or too close to me, and then it could adjust its scale in some sense. So it's basically, and this is in the brain, this is going to occur on a theta cycle, which is about anywhere between 4 and 10 hertz. So let's say 10 hertz. Every 10 times a second, essentially the location signal, location representation will sweep through a series of locations. And then along the line of movement, so that you could match an input early or late, and therefore know that your scale needs to be adjusted. That's a very high level description, and I can explain how it works. If anyone has questions about that.
Are you assuming the animal is moving along that arrow?
Well, I wasn't talking. I wasn't talking about the picture yet. So I was just describing the high level view that you essentially, we think about we're moving our finger or the animal, it doesn't matter, and we're updating the grid cells, the location as we move it. There's path integration going on, right? So as we move, it updates. But what actually will happen, it'll sweep through a series of locations along that line, closer and further away, and therefore you could match the feature early or late. And so if you match it early, then you know that the scale is, the thing is smaller than you observed it, and if you match it late, that means the scale is larger than you observed it. So that was just the overall top, and then I could describe what's going on here in this picture. Is that okay, or Jenny, you want to ask me a question?
Yeah, no, that's.
I do have one question.
Yeah.
Is that the only way you can get phase precession, is from a difference in scale?
That's the only way people have proposed that I'm aware of. No.
There's other ways.
Not difference in scale. Not difference in scale. I haven't talked about phase precession yet. I haven't even mentioned that yet. So I'll get that in a second, Kevin. I was just describing at a very high level view, because the actual details of how the neurons do this can be complicated. And I've struggled with understanding all the different theories about it. And I'm assuming not everyone here knows all that stuff. So I'm just trying to give a very high level view. What is the solution? It's going to end up basically saying, we're going to sweep along this. We're going to sweep the location along this over and over again, and we can match earlier or late. So let me just go through some of the details here. So in the upper left-hand corner, you see a typical image from a paper about grid cells. Ignore the arrow for the moment. And what you can see here is that these little things, I'll call them fried eggs. These are areas where a grid cell might respond in an environment. So this could be an animal moving in an environment, but it could also be our finger moving over an object, right? It's the same thing. So just imagine now this animal's moving in an environment, and as it goes along, this represents the response of an individual cell. So one cell would respond in each of these little highlighted circle areas. And they're quite large. You see, they're not very specific. And so they're color coded by basically on the frequency of firing in this case. So they fire more actively in the center of the little fried eggs as opposed to periphery. So there are two things going on here. There's a lot of theories about this, but all the theories, how grid cells come about are basically based on the same idea. And that's in the drawing below it. The drawing below it shows two sort of sine waves. The first one is labeled LFP, that stands for local field potential. This is a something called a theta hertz frequency. It's between 4 and 10 hertz. This is the frequency you observe in many parts of the brain or different interactions. It's like a frequency that's shared for different modalities, for example. So this would be shared in the hippocampus or the entorhinal cortex. So all the cells that's why it's called a field potential. It's a field that's all the cells are being subjected to. Then but the idea that people hypothesize, well, how do grid cells do this path integration, which by the way, they do in the dark. So the path integration isn't relying on any sensory input. The basic idea is that there's a second frequency. And where that frequency originates is a little bit controversial. Some people say it's in the dendrite. Some people say it's in the second cell. Doesn't really matter. There's a second, I'll just label that one. That's the red one. And it's called cell. And the frequency, that frequency is theta plus a small delta. So it's a little bit faster than theta, but only if this cell it's only if the animal is moving in a particular direction. All right. So this is sort of a frequency that says, well, if you're moving in a particular direction, if you're moving slowly, I'll increase the theta a little bit. If you're moving faster, I'll increase the theta even more. And so now the cell is getting two inputs. It's getting the LFP theta, which is sort of a constant, and it's getting this slightly faster frequency, which is related to the movement speed along a particular direction. And as you see, what happens is these two sine waves or these two frequencies go in and out of phase. And that's the black line, the third line down. The black line shows the sum of those two frequencies. And the idea here is that as an animal moves, let's say along the gray arrow above, and it's going through like multiple points where the cell's going to become active, what's happening is that cell is just sitting there and it's getting these two frequencies and it slowly goes in and out of phase as the animal moves. So again, that red sine wave is indicating the animal is moving in some direction at some speed. And if the animal is moving faster, these two cycles will go in and out of phase quicker. And that's as if the animal is moving quicker across the environment. And the animal is slower across the environment, they go in and out of phase slower. So this is the general belief how grid cells become active based on movement and not on sensory input. How path integration occurs, you have these frequencies that are going in and out of phase. Now, there's two things that are going on here. One is you could see that at first people say, well, there's a maximum firing rate of the cell, and that occurs at the peak of the black line there when the two cycles are in phase. But in reality, that's not a very good reliable indicator that where the cell is, because they make it look like, oh yeah, there's more of those little tick marks in the center and it gets fewer on the side. But in reality, it doesn't look that clean. But the second thing which is going on, which is very interesting, is you have this thing called phase precession. And what it means is that when the cell spikes, when the cell actually emits the spike, it's at different points in the phase of the theta frequency. So you can see those little dotted lines coming down from the troughs of the I guess it's blue sine wave at the top, and then you can see where those line up with the black line at the bottom, and where they line up changes over time. That is where the cells actually spike is moving from one part of the theta phase to another part of the theta phase. So if you look at actually when the spikes occur, you can tell where the animal is relative to that grid cell's ideal response pattern, right? And so as the animal moves towards a point, the grid cell fires. I think it fires early first. Yeah, there was a late first. Yeah, early first, and then it gets right on the trough of the LFP, the theta cycle, and then it moves beyond that. So there's sort of a shifting of when the spike actually occurs as the animal moves towards this center of its field and then beyond it. And this phase precession is seen in many cells in the brain. It's seen in grid cells, it's seen in place cells, it's seen in other cells. This seems to be a very common phenomenon. And what mostly people say is, oh, well, that's a good way for the brain to know, be more precise about where you are. So the grid cell says, yes, it's responding over those big, egg-like blobs. But if you look at the phase of when it's responding, it's much more accurate. You can say where the thing is. Okay. Any questions about that before I go on? Maybe if you had a question, Kevin, that would be a good one. I don't know if that was a good or good question.
You look at the fried egg where the center red piece is, that's going to correspond to the middle of
yeah, that corresponds to where my arrow is in the lower diagram.
Yeah, right here. Yeah. And then and so this entire thing you're showing here corresponds to one fried egg. It's not the entire trajectory of thing. And what you're saying here is, if you look here, this firing is near the beginning of the phase, and when it leaves the fried egg, or on the other end, it's sort of on the right hand side.
Yeah.
Of the, just in case that wasn't clear to people. I think one question for me, in the middle it shows multiple spikes, but my understanding is it's within a single cycle, you're not going to see multiple spikes. You're going to see one spike. It's just across multiple cycles, you're going to see more spikes on average.
I didn't know that. I had the impression that there'll be several spikes at those peaks.
Yeah, there would be several spikes. It's just my question is it like during a single cycle you're actually going to get multiple ones? But then
I think
the phase wouldn't be very precise, because it takes 5-10 milliseconds to do a spike.
Well, if we're at 10 hertz, that's the maximum sort of then you got 100 milliseconds.
Yeah. If you're going to do multiple spikes, that's like a 30% error.
Well, I think it's like 2 or 3 spikes. And I don't, they make it look here in that diagram like, oh yeah, there's clearly more spikes in the middle. But I think in reality, it's not like that. I think it's kind of noisy. There's a few spikes. If you look at the drawing on the right here, they're showing the same phenomenon, but I don't know. I think I didn't show a real diagram, like a real recording from cell, but I think if you look at a real recording from a cell, it's not so clean. It's kind of messy. But the point is that the phase precession seems to be pretty accurate, even though the spike count is not. So the idea is that really, they're saying, well, the accuracy comes about from the phase precession. And no one has any speculation, as far as I know, how the brain would use that. How would it take advantage of the fact that, oh, it's early or late on the phase to know something more precise? Well, I'm going to propose something in a second.
Okay, so as the animal is moving along that gray arrow in the upper left diagram, the phase is going to keep shifting back and forth as it gets to its preferred, each of the fried eggs.
Yeah, right.
Okay. As it's moving.
So if we can, we'll just go, oops, I just lost I tried to scroll the image over, now I lost it. Let's see if I can get it back. I'm doing this on my phone, it's not so easy. I lost the image.
You might be able to like swipe the whole screen to the
Oh,
left.
oh yeah,
right.
you're right. Yeah, you're right there. Yeah. All right. So on the right, so it just occurred to me there's something else to be happening. So on the right, I show two, our classic model, which is a location layer and a feature layer, right? This is from columns and columns plus, and so we're essentially saying, as a model consists of a series of locations and the features associated with them. And if your finger or your eye or your body was moving through space, what you would do is you would see a continuous series of locations and a continuous series of features. If you're moving your finger along the edge of an object, and so this goes back to my argument recently that you could do this type of model in a continuous fashion. But what occurred to me is that as you're moving through space, what you really would be doing is on each theta cycle, you'd be cycling through a series of locations, further away, where you actually are, and closer in. Because the phase precession thing, remember, we just looked at one cell before, but there's all these cells are doing this. And so there'll be cells that represent all these locations. And so at any point in time, you'd cycle through a series of locations. The ones that are spot on, where you're aligned with the peak of the phase, the theta, and ones where you're shifted early and shifted late. And so as you're moving along, on every theta cycle, you'd be going bum 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 on every theta cycle, cycling through a series of points along the line of movement. And so in some sense, it's saying, now you've got this feature up in the upper layer, and the feature's in there. We're trying to predict this feature. The feature's either there or it's coming in. We're trying to match it up with the location. If the feature is not in the right location, but it's within that sweep, it'll be found. It'll say like, oh yeah, I found it. There it is. I matched the feature I'm expecting at the location, but my location was either shifted to the left, or, shifted early or shifted late from what I expected. And so then you would say, yeah, I found it. I found what I was expecting, but it wasn't in the right place. It was shifted. And that's the information you need now to basically handle scale. And the way scale is handled in situations like this, or at least it's speculated to be, and I think this is right, is the way the brain does scale. It just changes the base theta frequency. If you increase it or decrease it, everything scales up. Everything, all the cells that are based on this methodology will scale up and scale down. So you could scale up and down movement. You could scale up and down, your path integration, motor behaviors, and so on. So now you have basically what you said. Okay, you're saying, okay, I'm expecting to find this feature at some peak activity on some grid cells, but I found it on an earlier or late phase activity of some grid cells. And I didn't try to work it out, but that's all the information you need to do now adjust the theta cycle to match what you actually found. If I found that I was early on the cycle, I would have to speed up the theta cycle a little bit, and now I'd be matching it. So the end goal of all this is just adjust theta, which essentially adjusts the scale. And by noticing that I was some amount off of what I should be, it gives you the information to say move the theta to match what I actually observed. So that's the entire idea. I can think of some potential problems with it, but this is it felt like this is going to happen whether we wanted it to or not. This didn't require any speculation on my part. This is just what grid cells would do. And that's why I said I stumbled upon it, because I wasn't trying to solve this particular problem. Again, I don't think we have to do it this way necessarily. It's just the idea is that you sweep through a series of locations along your line of movement, and you see which one matches better. And when you find the one that matches better, then you have the knowledge on how to scale the system.
Does it matter how fast you're moving?
Well, the faster you move, the greater if on the left, the greater the frequency of the red oscillation.
Right.
Right. So right, it does, because if I was a cell, imagine I'm a cell, I'm just going in and out of phase, and every time I'm going in and out of phase here, I mean not phase, I'm going in, I'm either my red oscillation is matching the blue one or not. And every time it matches the blue one, I fire. And if my red one is going faster, then I will do this. I will cycle through these on and off patterns faster. It's like if I'm moving faster, then I will go through my locations faster. So it does matter how fast you're moving, and that has been shown to be the case too. That this the red oscillation is velocity dependent.
So the LFP theta, is that global across any set of grid cells, or is the adjustments to theta, which you're proposing as a accommodation mechanism, can you have more than one theta going on in the brain?
Yeah, more than one theta in the brain for certain. I don't know how global it is. Clearly, what's going on in your visual system is different than what's going on in your tactile system, or maybe not. But my understanding is that it's global to some region, but not the whole brain. That's my understanding about it. But we could look that up. I haven't read about that in a long time, so I'm not sure. But that's what I kind of distilled.
So the other question I have, when you mentioned cell 1, cell 2, cell 3, if I were to superimpose the Friday diagrams of cell 1, cell 2, cell 3 on top of each other, what would that look like? Are they just displaced just a little bit, or how do those quote unquote receptive fields look?
Right. In fact, in that diagram, you can look at, like, there's a little highlighted area in the middle, right? I didn't make this diagram. I just stole it from someplace. That's basically saying at some point in movement, so this 1 second, that means the animal is moving, right? The animal is moving, and this is what happened over the second of movement. At that point, in the animal is at some location, and you see first you get a red cell, then you get a green cell, then you get a blue cell. This is the idea that you're cycling through 3 locations very quickly as the animal moves, and they're happening very quickly. Of course, you're just separated by some distance in the phase of the theta oscillation. So those are like overlapping eggs, if you will, right? Of course, when we looked at the egg diagram, that's just one cell. There's cells representing every other point on that blue background. So there's all these overlapping eggs, in some sense, lots of them. There's no point in that space that's not represented by grid cells, and they're overlapping quite a bit, actually. So
okay. So what I'm seeing is that by tapping multiple of these cells and assuming they're in some kind of communication with each other, you potentially have a vernier mechanism that refines the phase accuracy against the more cells you can harness for that, the phase differences, I think, become more accurate.
Maybe I didn't follow that.
Like how
is that necessary?
Is it necessary? I think in the sense of it right now, the bound of accuracy is on how finely these cells' respective fields are placed. But if you allow them to, if in some sense, interfere with each other, you can probably get a finer resolution out of that. I think something like that occurs separately from this in the retina, where you actually get a finer level of detail than the actual sample frequency you would suspect from where the retinal receptors are. So I'm just proposing it as a mechanism that there actually might be a finer level of control if it was necessary by harnessing multiple of these things.
Yeah, that's possible. I don't know. I think I'm just looking at it pretty simplistically. Imagine that I'm looking at the red, green, and the blue cells firing succession in that highlighted area. What we're just basically what it's saying is that's, I don't know if I need more resolution than that. But basically what's being sent to the upper layer cells, to the feature layer in the upper diagram, it's saying, okay, does this location match your feature? Here's a location. Is that matching what feature we're having? And then here's another location. Does that match what feature we're having? Here's another location. Does that match feature we're having? And I don't know if I need finer granularity than that, but maybe it would be helpful. I don't know.
Well,
but I
I'm
wasn't relying on that. I just said, yeah, it should work like this.
I'm thinking in the case where if you want to harness this thing for something like curvature or something, and you want to be able to detect multiple curvatures, you know, in parallel, each little group of cell could.
Why would you want to take multiple curvatures? What do you mean by take multiple curvatures in parallel? I don't know what that means.
If you want
also,
to
I think curvature would be done in a different way. You wouldn't use detailed locations to detect curvature. You just get the curvature directly from the spin.
Well, you detect curvature in the feature layer.
Yeah,
but I'm assigning the curvature to the location, right?
Yeah.
Okay.
Right. Oh, I see, Kevin. You're saying if you want to get a finer resolution of curvature. Yeah, I wasn't even thinking about that at all here. I'm just assuming that you've got a representation of curvature in the upper layer, and it says I know what location I'm supposed to be at, and yeah.
Okay.
But you can imagine how this thing would get synced up pretty easily. Essentially, imagine if the red cell matched the feature, then I'd say, oh well, now if I just try to synchronize the theta with that new peak, if I just say, okay, let's move the peak of the theta cycle, the LPF LFP one, to the where the red cell actually fired, that might be sufficient to set the new theta. Basically, every moment you can say, I should just try to sync up the theta onto the, when I have a match, I should try to sync the theta up to that point. This is an additional point. I'm not reacting to what you said, Kevin. So I think there's very simple.
That's fine. That's fine. I'm just trying to extrapolate on it to, say how, if Subutai basically saying that says that he thinks curvature is actually something else, I'm trying to think of the notion of scale invariance as applied to the problem of curvature.
I see. No, I wasn't doing that. This is just scale invariance in terms of the reference frame made out of grid cells. If I can get it to scale, the challenge that we were facing is I have a model I learned at some scale, and now I'm observing an object that's at a different scale, and so the model doesn't fit anymore, right? If I move a certain amount, I'm not going to see the right feature,
right?
And therefore I won't recognize it. And all this says within a certain amount of variation, this system would be able to quickly detect, yeah, it's right, but it's just off a bit, and now let's adjust it so it's correct.
So one question I have, so in order for this to work, cell 1, cell 2, and cell 3 all have to be representing the same location in the object's reference frame, right? It's just slightly, it's at a slightly different scale, right?
No, that's not right. They're at slightly different spacing. Cell 1, cell 2, and cell 3 are overlapping eggs. They're basically saying, they're not at different scales. They're not at different scales. They're just different locations.
Yeah. Okay. So they're different locations in the object's reference frame.
Right.
So if you're looking for a feature, and the object is smaller or larger, not quite sure how this would actually work. Would this actually work?
So all we're trying to do, recognition in our models is basically finding or doing a correct prediction or being synced on a model is basically matching a location and feature at the right locations.
Right.
And all this is saying is as you're moving, what would naturally happen is you're sort of, in some sense, trying expanded and contracted spaces 10 times a second. You say, does any, expanded space, contracted space, expanded space, contracted space, does any of this stuff match? It's like, you're not actually expanding and contracting space. You're just essentially, you're trying different locations along the line of movement
No, I get that. I get that.
Okay.
Yeah, it's just that in our model, the feature is associated with a particular location in the object's reference frame. And we don't know what the scaling is between the sensor movement and the change in the object's reference frame, right?
Well, we do know, we know based on the path integration methods that if the scale didn't change, then it's all set, right?
Yeah, but we're trying to determine what the scale, we don't know what the scale is there.
Right.
And so the change, if you move, you know, 1 inch, if you move your finger 1 inch, the actual change in the object's reference frame could vary depending on the actual scale of the object.
Right.
Right. And so, but in our model, it's the object-centered object-centric location is associated with each feature. So there's going to be, so the location represented by cell 1 would be associated with that feature. Cell 2 and cell 3 would not have an association with that feature, right? Because they're different points in the object reference frame.
Right, right. So we're going to find out which of these series of locations matches up with the feature.
But none of them can. Only cell 1 can, regardless of the scale. Because, because cell 2 and cell 3 represent different points in the object reference frame. They're never going to match the feature. You see what I mean?
Well,
because the feature is at a specific location in the object reference frame.
Right, right. But so what we're basically saying, we don't know our location. We're not certain. What the cycling up through these locations says, we're not really certain where we are. So let's see who matches up. Only one will match up a particular feature. If that's what you're saying, I agree. Only one of those cells will match up a particular feature. The other one will match up with a different feature, and another one will match up with a different feature.
Right.
Right. But you didn't see how it would work, so I'm confused by that.
Yeah, I'm trying to think through step by step. So let's say the red cell is associated with the tip of my pen. But the pen happens to be larger, longer or something. And so, you know, it's expecting, as I move my finger, it's expecting the tip. It's not going to get the tip.
Well, let's say the following. Can we say that instead of the red cell, let's say the green cell is the tip of your finger.
Okay. Yeah, the green cell is the tip. Yeah.
Right. So if we were on spot on, meaning the model and the thing I'm inferring right now are the same scale, then when the green cell gets to its when it's at the at its center of its egg, it's going to match up to that feature.
Right.
Now, if let's say the pen is longer now or shorter, what'll happen is the green cell won't be the one in the middle here anymore. It'll be one of the shifted to the left or shifted to the right, because I'm not at the tip. I'm either before the tip or I'm after the tip. And so the green cell would match the tip, but it wouldn't be the center cell now. It'd be like the red cell or the blue cell. Mm-hmm. It would be saying, yeah, I got the tip, but I'm offset from where there's a phase shift here. So
okay, so we need some mechanism to determine what that offset is to adjust the scale. Somehow.
Well, no, it happens automatically. Well, are you following me on that?
Yeah, I follow. Yeah, I follow that now.
So at this point, imagine you were the red cell, and you maybe that at this point you matched your feature, but you were offset on the phase. So we now know that a shift is required, like a scaling is required. We know that because the red was not at the peak. The red was on the edge of its egg.
Yeah, but what's the mechanism for that?
Well, I don't know, but I kind of suggested one a moment ago. You've got the LFP, which is the black line at the bottom, and now you have we're actually trying to match up to the trough of the LFP, not the peak. The trough is the one here because this is depolarization. Yeah. And so ideally, your cell would have been matching up with the trough of the LFP. But now you're not matching up with the trough. Like on the left here, if you're the red cell, you're sort of matching up more towards closer to the opposite, the peak. And so now imagine if I said, okay, at this point, what we're going to do is we're just going to force the theta cycle, the theta rhythm, to bring its trough in line with the cell firing. So I'm just going to force the theta cycle to sort of, I don't know how I would do this, but it seems pretty simple idea that you say, okay, we should have been lined up. So you line up with me right now. I'm going to reset your cycle to your frequency to match up with me. And so it's just the fact that the two peaks aren't aligned. It's the cell is
yeah,
firing,
I get that. I was just wondering what the actual
I don't know what the actual mechanism is.
But it would be the arrow from the feature layer down to the location layer, that there would be information there somehow that.
Yeah, one can imagine that maybe it's just speculating. I mean, the information is there, right? The information is there very locally. The information is there sufficiently to say, you know what, just line these two things up, try to get them to be in sync. And how that happens, I don't know. I mean, I think in this case, in the entorhinal cortex, I think the theta rhythm is established between between the reticular formation and the cortex? No, I'm in the cortex. I don't know enough about it. I suppose we could look into it, but from an engineering point of view, it's not that hard to imagine how to do this. From a biochemistry and biophysical point of view, it might be trickier.
So,
but
Jeff, I have a couple of basic questions here. I don't know if I'm the only one who's about 2x behind you, but I'm just struggling to keep up here. Can I ask some basics? I
warned you. I said the biology in this area is really complicated. So.
Well, I have no problem working through it over a long time, but I'm behind right now. So cell 1, cell 2, cell 3. First question: Are these responsive to slightly different locations, but similar locations?
Right. They're overlapping eggs. Yes.
Okay, so these are overlapping eggs. My next question is, they share this common LFP input? That's input across all cells.
That's right. That's right.
Okay, and we're going to just assume for now that those synaptic inputs from that LFP, that's from some other population of neurons. That's not other neighboring grid cells. Is that right?
That's right.
Okay,
and the LFP could be in that same population. It's just sort of a global mean of all the firing.
Right, but
is the LFP a traveling wave, or is it static?
I don't think the LFP is coming from grid cells. I don't think people think the LFP is coming from grid cells.
Well, LFP is whatever they measure, wherever they put the probe to measure.
Well, all right. The theta frequency is generally believed to be generated someplace else. We could go look it up. I think there's a theta frequency in the cortex, and I believe it's being generated between thalamus and cortex. That is somehow the interaction with the.
That could be the source of it, but aren't they actually measuring all of the cells in that same region? That maybe that's the exact source of it. Yeah, so all of those cells are going up and down based as a global.
When they do this, the problem is they're measuring the voltage outside of a cell.
Right.
That's why it's a local field potential. It's not inside of a cell, and so it is an accumulation of a lot of stuff.
Yeah.
It's hard. You can't really put your finger on exactly where it's coming from. And so I didn't even try to think about that.
Yeah, that was going to be another question is like, are these LFP inputs to a cell? Does it really look like this, or is, really a cell should be receiving spikes? So like, what is the signal? Do we know what the signal to a single one of these cells looks like?
So there's a couple of theories about this. If you go look on the left side where I showed just the red and the green arrow, the green sine waves.
Okay.
You don't have colors.
Red and blue sine waves.
Red and blue. I'm sorry, red and blue. Yeah. There's a couple of theories. There's two theories about where the red cycle's coming from. One is that actual individual dendrites have their own oscillation. and that they're oscillating, that's the red oscillation, and that there are synapses coming from other cells someplace that are providing the theta oscillation, and that's closer to the soma of the cell body. So you have the blue thing sort of driving the cell body, and you have the red thing coming from a dendrite. That's one theory. And so the dendrite itself has its own intrinsic oscillation, and it's interfering with the synapses that are coming from someplace else, which represent the LFP in this case, but you can think of it as synapses on the near the soma. So you got like two synaptic inputs, one coming from two depolarizations, one coming from a dendrite and one coming from synapses from someplace else. That's one theory. You follow that one, Ben?
Roughly, yes.
Yeah, it's confusing. I don't like that one.
Well, is it, I guess, the other question is, are just the LFP inputs enough to make the cells fire alone or not? It looks like on the diagram on the left, if it's not synchronized, it's not enough.
No.
So
No, the cell has to detect a pattern of inputs coming in to fire. It's not just the global.
Right. Otherwise, every grid cell would be firing it all the time.
okay, so we have a sub-threshold oscillation going on.
Yes.
Okay.
Yeah.
And one hypothesis is that inputs to the dendrites are responsible for the red oscillation.
That's one hypothesis. I don't like that one, but that's the one I read a paper this morning. They were still proposing that one. That has some real problems to it. Another possibility is there's a set of cells that are firing like the red guys at different phases, and those other cells are providing synapses. So in that case, it'd be two sets of synapses on a cell, one coming from a population of cells that represent the red oscillation, and one set of population that represents the blue oscillation. And they're just synaptic inputs to the cell.
So I have a question. So the LFP, I mean, could either be static in the sense that everyone's getting the same signal, synchronously, or it could be a traveling wave where it moves across the grid cell region. There's
neither.
different models.
Neither. It's neither.
Neither?
No.
So how can it be neither?
It's just the global average of firing in the cells in that area. It's not that every cell is getting this exact input. Every cell is detecting some other pattern, but this is the global average of the cells firing up, going up and down.
Well,
But any individual cell is not getting this signal.
I thought all the grid cells would be getting the blue signal.
Yeah, the blue signal. Oh,
yeah.
Which blue signal?
That's what I was saying. That's what I was talking about, the LFP, the blue signal. So, I mean,
I don't think it's not a single signal. It's just a global average of a bunch of cells firing. I don't think.
Well, if it's a global average and they're phase indiscriminate amongst themselves, then you don't have a mechanism at all.
No, they're not.
It's not even.
They are in phase. It's just that some smaller number of cells will fire in the middle, and a larger number will fire in the peak. But it's not like there's a signal coming in that's up and down like that. It's a pattern of cells. It's an SDR of cells.
Well, but I think isn't it true that the LFP is experienced by all the cells in this area?
I think all of the cells in the area, I think all you can say is that the mean of all of the cells in that area exhibit that cycle.
Yeah, but the
That's
assumption
not the same as every single cell getting it.
Well, the assumption is that all the grid cells in a grid cell module would be experiencing the same LFP.
I think they're exhibiting the same LFP.
Well,
does it make sense to think of all the cells kind of like sampling from that LFP? So like two neurons, they're going to have slightly different versions of that blue curve and maybe like distorted versions of it, but maybe on average they're similar. Just trying to kind of get the right cartoon image for myself.
Well, but here's the thing. My understanding of all the grid cell theories assumes that all the grid cells in a particular grid cell module are, if you measured outside of them, they would all have the same blue oscillation. It's just remember, it's an oscillation in voltage. In fact, if you think about it, a cell fires when it's a differential between the inside voltage and the outside voltage, which matters. It's not the absolute voltage, it's the differential. And so even just the fact that you have some sort of potential voltage that's oscillating outside, you don't even need to have synapses. It's just the cells will more likely fire at the trough of that, or the peak of whatever. I have to think about it. Look, I bet you this, maybe Supertel, you know more about this. I don't know a lot about this LFP, but it seems, I'm not sure if these are essential issues. I think the important thing is, maybe we disagree about this. It seems that all the grid cells in a module have to be experiencing the same blue oscillation.
Yeah, I don't think it's essential. I don't think it's essential at all. All we care about is that these cells fire in this phase relative to this.
Well, the theories I've read about, which makes sense to me, which I've kind of absorbed immediately and say, oh, yeah, that's gotta be right, is that you could speed up and slow down everybody simultaneously by just adjusting this LFP, this global LFP. Yeah, you could scale the entire object. That means everybody has to be experiencing the same LFP. It's
no.
Well, how not? If it was
because it's just a ensemble of cells that are sending input into that region. Their firing rates would have to speed up or slow down, but it's not like there's a single scalar value that everyone's receiving that has the LFP. You see what I mean?
Oh, okay. I think I see what you mean. Is that a difference that makes a difference?
No, I don't think. I think it's purely an implementation detail. I don't think it makes any difference.
Okay.
What you're proposing. I'm just trying to react to what Kevin was saying.
All right. I see.
Well, okay.
Well, I don't think it's really important for today, right?
No, it's not. No.
Today, the idea is really simple idea, is that we don't know the right scale. So as we're moving along and we're predicting the next location, we'll sweep through a series of locations in that direction and see if any of them match. And if one of them matches early or late, then we say that's good enough and we'll just adjust our scale and keep going. So there's a lot of ways we could do this in software.
Right.
And I certainly wouldn't do this the way they do it in neurons. This is really
in software, it's way easier.
Yeah, I was just thinking this way, right? For example, think about a temporal memory of a bunch of locations. Can you see the dots? Which are, let's say, these are possible locations where I could be at this moment. And let's say I know the direction. Let's say I go this way. Then essentially, for each of those dots, sorry, I don't see my mouse. So I would have a whole sequence of possible values where I could be, depending on scale, right?
Yeah, and I see which one matches my model.
And one of them will match, yeah.
Well, if it's the scale version of a model I'm tracking, then one of them will match, right? And then, by the way, what if imagine I had an object which had two features that were really close together, and another object that had the same two features a little further apart? I might get confused, but I wouldn't. But you have to continue inferring, right? As you're going along, just because you found one feature that's the right, it's off by a scale, then you're going to keep trying, and then you might find the next features aren't. So you can't guarantee that when you find a single feature matching this way that you've discovered the scale of the object, because it could be another object that just has the same features closer together or further apart. So but I think in the end it would all work out.
Yeah, right. Yeah, I think the tricky thing is that there's a lot of possibilities, because I could essentially, from any of those start points, I need to check all those possible places where the next feature essentially could be. So,
yeah, but it clearly the brain's doing this, right? Because it has to do it on like a 10 hertz cycle. So it's got plenty of time to cycle through a series of locations. And see if
the reason I was asking a question was that, I was trying to hypothesize that there's a self-timing operation where the theta cycle originates from the interactions of cells themselves. And another way of doing it is as a traveling wave. If you imagine there's a traveling wave across the grid cells in the direction of motion, a lot of things become possible.
I don't think it's going to be I think it's unlikely to be the traveling wave, Kevin.
But has anyone measured it and said that?
There are traveling waves in the brain, but again, think about it this way. In the cortex, it's believed that the theta cycle originates in the thalamus, and then the thalamus broadcasts broadly to all different areas of cortex, right? So there wouldn't be that's not conducive to doing a traveling wave. That's more like a broadcast signal. And so
But there's
I
a
wouldn't make sense as
propagation
traveling.
delay on doing that.
Yeah, but imagine the
And-
thalamus is sort of centrally located, and the distance between the thalamus and like all areas of V1, for example, is that distance. You think of it more like uniform as opposed to it just if I'm broadcasting from some central place, I can't really have a traveling wave at the place I'm broadcasting to. A traveling wave requires
Okay, if the radius is all the same, then I agree with you.
But even if it's not the
If
same,
the
it wouldn't
radius
be a
is all the
traveling
same, then I
wave.
agree with
It
you.
might not even reach the same areas, but it wouldn't. A traveling wave essentially requires like the wave in a stadium, right? It requires people to wait till the guy before them happens, right? In this case, a traveling wave would say, I'm going to go when my neighbor goes, and then someone says, I'm going to go when you go. But in this case, it wouldn't be that way. It might look like a traveling wave, but it wouldn't be a traveling wave because it's being broadcast from someplace else, and it might be just arriving at different time frames. So that's why I said it wouldn't be a traveling wave. It doesn't mean it exactly has to happen everywhere at the same point, but it's not a propagating wave reliant on other people, other neurons nearby.
Okay, so the argument is there is from the geometry of the brain. You're saying that it couldn't be a travel or doesn't need to be a traveling wave because
well, it's more like
propagation delays. It's going to be the same to all.
Yeah,
these points.
geometry, or more, it's saying if the theta cycle is really being generated, let's say in the interaction between the non-reticular formation in the thalamus and the cortex, that's a sort of a single generator, if you will, and therefore if you have a single generator, then it's not a traveling wave. Right? Traveling wave says you start at some place and it sweeps across. There's a lot of those in the brain, but at least in the retina, there's traveling waves. I don't think you need that here. I think the simplest explanation, you just say, okay, just assume we have a theta, or we have a scale that we're shooting for, and we'll sweep through different scales as we go along. If you follow all this, it also made me wonder if we could solve some of the other problems this way, like the problem of orientation. I could sweep through different orientations. Now you guys have an alternate solution.
Yeah, yes, it's sort of, it's similar, but instead of sweeping, I think it's effectively might be very similar. So sweeping through different orientations is effectively you try different cortical columns at each of their orientation, and this way you narrow it down, you down select, and then at some point just
do we have time?
So we can go through that tomorrow.
We can do that tomorrow. Yeah.
Yeah,
okay.
that would be it. Take that. I mean, we didn't have this detailed mechanism. In fact, I don't think we have a biophysical mechanism yet.
No.
Well, I was just curious what the general idea was. Can you, in the next 10 minutes, can you give me a general idea what you're thinking?
Yeah, sure. It might help. I show some visuals.
I only have 10 minutes.
Yeah, 10 minutes. Okay, I think that's fine. So let me share.
I mean, I'm sure it'll take more than 10 minutes. So maybe you can just give me like something to think about.
Yeah, I can give you an overview in 10 minutes. That's fine. All right. So yeah, so that's essentially the issue. Let's say I have the cup, and I move along a cup with a displacement red arrow, and I don't know what is the orientation of the cup. So a movement a displacement that I observe, here the red arrow, could be in the coordinates frame of the cup, could be any of those directions. Right?
Yeah, I got it.
And so how could this be resolved? And here, thinking about the temporal memory in the columns plus paper, so we have associations between the sensory layer and the location layer, and we narrow, in each step, we narrow down the union of possible locations and the union of possible sensations. Now, the difference between the columns plus paper is we have now a whole set of those modules or cortical columns, and each represents has its own orientation of the cup. So when I now do a displacement.
Are those multiple grid cell modules? Is that like 4 grid cell modules there?
Yeah, each would have its own grid cell, and yeah, let's say this. If this is grid cells, it could be other location code. But if this is a grid cell, it could be the same as the columns bus paper. This you have multiple.
So I'm just saying there's multiple. Those 4 blocks
Yeah.
are like four separate
Yeah.
path integration modules.
Yeah.
Okay.
Yeah. So it would be exactly the same. The only difference is the way it's stacked, like from the top to bottom. So you have 3 of those, and then each would represent a certain orientation, and with the orientation, essentially its own transformation of the displacement to a certain direction on that place code. So if it's in that orientation, the director, it would go like this, and if it's that orientation, it would go like that, and so on.
we have basically we have multiple guesses on what the orientation is.
Yeah, we have multiple guesses.
multiple guesses.
Each starts with a guess. And then essentially, as a group, they narrow down the possibilities. Because then, let's say initially you might be at all those red locations. Then you check. This guy would check. Oh, is the feature at those locations here where the arrows point to?
So if I have a set of guesses,
Mhm.
some are going to be more correct than others,
Mhm.
and then the correct ones inform the other guys.
Yeah. First of all, those are wrong. Let's say all this. Let's say the feature I observed wasn't over there and wasn't on this side. So I know these wouldn't be possible anymore. Then this would be the only column surviving with activity. So the activity would like down select, and you would end up just at the end with one of them, and then the other columns that could possibly be reused with a new guess of a location. And
so one couldn't they inform each other?
yeah, they could inform each other.
Yeah, no, they would. Once one of them matches, you somehow broadcast this, and they would all then go into the same.
Well, you know, like if you think about grid cells, they look at multiple grid cell modules in like the entorhinal cortex. They anchor independently, meaning like I go into a new environment, they'll anchor which cells are active, and it sort of like looks like it's random in some sense. But once you've learned an object, they're always the same, and that is they all anchor together. So which would imply that there's a learning there, like they socially link to each other somehow, saying, yeah, okay, well, you know, my point is that they're not always linked, but when in the learned environment they are linked. So you could just imagine that when one guy says, I'm working, it's working for me, my orientation is pretty good, then that could inform the other people right away. They could say, well, okay, well, if that's the case, then I know what I should be. Something like that. It wouldn't have to. It wouldn't have to be random.
Right. Yeah, that's good. And then in this way, if you need to refine your location, because you don't want to spend like thousands of columns just to get an accurate direction, you could just have a small number, let's say 8. And then once one guy says, oh, it's in that direction, and the other guys can use the same information and on its own have a slightly more refined guess. And in this way, you improve the accuracy as you go, while not requiring a lot of columns devoted to figuring out what is the object and orientation.
So it sounds
like I think what you're saying, though, this may be a language issue, but you may be saying something different than what we discussed the other day. It's like use multiple modules, one for each orientation guess. I don't think we discussed that. I think each row here would be a different orientation guess, right?
Yeah, each row would be a different orientation.
Yeah, but each row contains multiple modules.
Oh.
You see what I mean? So
that's a lot of grid cell modules then. Why does it have to be that way, Subutai? Why couldn't each block in a row have its own orientation? Or because I was assuming those are each a grid cell module.
They are each, yeah. Each one is each of them are their own grid cell module. But I think what we were thinking is this row would have an orientation guess. This row would have another orientation guess.
Right.
This row would have a third orientation guess.
Yeah, that's right.
Each layer would have. So we would use multiple columns, not multiple modules.
Yeah, I meant modules in our sense of the Monty system.
Yeah. No, that's what I mean. language issue here. Modules is a ambiguous term here. So,
So-
what Jeff was asking is that are each of these modules, and they are, they're each square is a grid cell module.
You
Yeah.
assume that they were in the same, you assume though those four were in the same cortical column?
Yes,
Yes.
just like our column.
Yes.
Yeah, what if that wasn't true? I mean, this is something that's really puzzling me forever and ever and ever, is like, okay, the best solution we've ever come up with is having multiple grid cell modules, but it doesn't look like there's enough to have multiple ones inside a single cortical column.
Okay.
And so could there be one grid cell module in each cortical column and still they be doing this voting across columns?
Yeah, I think this proposal is kind of independent of that.
Yeah.
There's the question of, okay, how do you accurately represent location within a column? Do you use single modules or multiple modules? But this, like, regardless of how you do that, this proposal is you have multiple guesses going on simultaneously.
But they could
and
be in
then-
multiple, they could be in multiple columns, right?
Yeah,
That's
we
how
have
you
to figure
just-
out the exact biophysical interpretation. Are there multiple columns, or could it? Ideally, it would somehow we could do it all within one cortical column itself.
Ideally, but there's a lot of cross, and it could be related to that. Of course, it has to
Yeah.
work with us. Well, okay, I just leave it at that. So
Yeah,
if I
let's
was
go through this in more detail tomorrow.
Yeah,
I think there's
I-
I think you missed a step here also, Heiko, that might help.
Okay.
Of how the movement is translated into these shifts.
Yeah, that's the same as in the columns plus paper. Yeah. So you would
Yeah,
learn.
but it wasn't, we didn't state it explicitly.
Yeah.
So Jeff, I think for Jeff and others, it might be helpful.
Okay.
Okay, that would be helpful, and that's good because I probably have to run here.
Okay. Good.
All right, well, it gives you something to think about. Thanks for doing that.
Yeah.